In this segment, we will discuss another important part of a control. And this is the post-translational control of protein stability. Once the proteins are produced, what are the controls which are there to control their stability, to control their function, to control their presence? Now, we know that a cell must possess mechanism to control the length of time for which specific protein they survive once they are fully functional. Now, the proteins like enzymes and other proteins, once they are produced, they are fully functional, then the cell must also keep an eye on them, that how long they can be there, how long they can have those specific proteins should function or remain in the cell. Now, then important part in this process which they play is a proteasomes. Now, what proteasomes do? That is the degradation of cellular protein is carried out within hollow cylindrical protein degrading machines which are called proteasomes. So, they, as a matter of fact, degrade those proteins which are there, and they are found in both the nucleus as well as in the cytosol of the cell. You see, because in the nucleus there are important proteins, and at the same time in the cytosol there are proteins which are present, so they should be present in there, so the unlikely or unwanted protein, they can be degraded. Now, these proteasomes, they consist of 4 rings of polypeptides, subunits, and they are stacked one on the top of the other with a cap attached at either end of the stack. So you can see a cap is attached on the either end of the stack, and there are 4 subunits of polypeptides in these proteasomes. The active sites of these subunits, they face the enclosed central chamber where proteolytic digestion can occur in a protected environment. So, in a protected environment, these subunits, they are enclosed toward the central chamber, which is the center of these proteasomes, and here the digestion can occur. Now, proteasome digest proteins that have been specially selected and marked for destruction. Now, the cell, it selects the proteins, it marks them, and then proteasomes are allowed to digest them. It's not that the proteasomes are there in the cytosol or in the nucleus, and they go around, and whatever protein they found, they just degrade it. No. They select those cell has to first make a decision and select the protein which are to be destroyed. Now, these selected proteins, they are recognized as abnormal, either misfolded or incorrectly associated with other proteins. So those proteins which are abnormal proteins, which are misfolded, which are not useful, so cell selects them very wisely and send them to the proteasome. The selection of normal protein for our proteasome destruction is based on the protein's biological activity. The normal proteins, how they are destroyed, so that will be selected on their biological stability, that how stable they are biological, so that they are useful for the biosystem. Now, longevity of proteins, it depends on their function. And every protein is thought to have characteristic longevity. For example, some protein molecules, such as the enzyme of glycolysis, metabolic, or the globin molecules of an erythrocyte, are present for days to weeks, because these are the metabolic processes and they are non-regulatory enzymes which are there. Other proteins that are required for specific fleeting activity, such as regulatory protein that initiate DNA replication or trigger the cell division, that may survive only for a few minutes. So regulatory enzyme or regulatory protein, they are needed for only few minutes. So this is the one which are to be destroyed. The destruction of such key regulatory proteins by proteasomes, it plays a crucial role in the progression of cellular process. They are very important that the cell has to destroy those proteins so that they can not be there, and the regulation of the cell, it continues irregular for time. Now, the factors that control a protein's lifetime are not well understood. One of the, as we have described earlier, that we know that some of the glycolysis protein or the regulatory protein, they can be there for a long time, but the controlling protein which control the nucleus or DNA synthesis, extra, they have to be destroyed. One of the determinants is the specific amino acid that resides at the end terminal of a polypeptide chain. So one, although there are many factors, but one is a specific amino acid residing at a terminal polypeptide chain helps the cell to decide that which protein is to be destroyed, and the polypeptide that terminates in arginine or lysine, for example, are typically short-lived. We have seen that the polypeptide containing lysine or arginine, they are typically short-lived and marked for that. So ubiquitin helps in a protein degradation. That's another. You know, the ubiquitin means everywhere. So ubiquitin is a small 8.5 kilodalton regulatory protein that has been found in almost all tissues for eukaryotic organisms. And in this case, you can see that there is ubiquitin and the proteasomes which are there. They are dragged inside the ubiquitin and that is destroyed, that is degraded into the combining amino acid, or further degradation is there into this. Proteins are targeted for destruction by attachment of number of ubiquitin molecules. Once it is polyubiquitinated, a protein is recognized by the cap of the proteasome. The cap, it recognizes the proteasome, which removes the ubiquitin chain off and unfold the target protein using the energy which is provided by the ATP hydrolysis, and the protein is destroyed. So this is, you see, how very interesting mechanism is there, that unwanted proteins, they are destroyed. The cell gets rid of that and regulates the proteins. Thank you.